4.5 billion years ago, while Earth was still a molten hellscape, life somehow appeared in just 600 million years. Scientists call this impossibly fast. We believe life crawled from primordial soup, evolved slowly, and eventually created us. But what if we got it backwards? What if life rained down from space, carrying the genetic blueprints of civilizations that existed billions of years before Earth even formed? If you're ready to question everything you know about human origins, hit that like button and subscribe. We're about to uncover evidence that we might be the aliens. Prepare yourselves, we begin. Picture trying to assemble a working smartphone by shaking a box of electronic parts for 600 million years. The math behind life's emergence on Earth breaks every rule of probability. When our planet finally cooled enough to support liquid water, something extraordinary happened within what scientists consider a cosmic blink. Complex molecules started organizing themselves into self-replicating systems. These weren't just simple chains of carbon and hydrogen. We're talking about intricate biological machines capable of copying themselves with stunning precision. Think about this timeline. Earth formed 4.5 billion years ago. For the first 800 million years, our world was basically a giant lava lamp, constantly bombarded by asteroids and comets. The surface temperature reached 2,000 degrees Fahrenheit. Nothing could survive. But somehow, by 3.8 billion years ago, we find evidence of complex microbial life already thriving in the fossil record. That gives evolution just 600 million years to go from sterile rock to living organisms. Charles Darwin himself called this rapid appearance the warm little pond problem. He knew that natural selection needed time, vast amounts of time, to create even the simplest life forms. Yet here on Earth, life appeared almost immediately after conditions became suitable. Consider the complexity we're dealing with. Even the most basic bacteria contains roughly 4 million individual parts working in perfect harmony. DNA, RNA, proteins, cell membranes, energy production systems. It's like finding a fully assembled Boeing 747 in a junkyard, where only raw materials existed the day before. Modern scientists have tried recreating these conditions in laboratories. They've mixed all the right chemicals, applied electrical charges to simulate lightning, and waited. The results? A few simple amino acids, the building blocks of proteins. Nothing even close to a living cell. The gap between chemistry and biology remains as mysterious today as it was in Darwin's time. But there's another explanation that makes the timeline make sense. What if life didn't start from scratch on Earth? What if it arrived here already assembled, already tested, already proven to work? Imagine life as a cosmic inheritance, passed down from civilizations that figured out the biological puzzle billions of years before our solar system even existed. This isn't science fiction anymore. NASA has found organic compounds throughout the galaxy. The European Space Agency has detected complex molecules in interstellar clouds. The building blocks of life aren't rare cosmic accidents. They're everywhere we look. The Panspermia hypothesis suggests that life spreads throughout the universe like seeds on the wind. Some seeds land on suitable planets and take root. Others drift through space for millions of years, waiting for the right conditions. From this perspective, Earth wasn't the birthplace of life. It was just another garden where cosmic seeds finally found fertile ground. If this theory holds true, then every living thing on our planet, including us, carries the genetic memory of civilizations that existed long before Earth was even a glimmer in a stellar nursery. But the evidence goes far deeper than just mathematical probability. Hidden inside the rocks that constantly rain down on our planet, scientists have found something that challenges everything we thought we knew about life's origins. Every single day, Earth gets hit by 100 tons of space debris. Most of these cosmic visitors burn up in our atmosphere, creating the shooting stars we wish upon. But some survive the journey, crashing into our planet as meteorites. For over a century, scientists have been cracking these space rocks open, expecting to find minerals and metals. Instead, they've discovered something far more extraordinary. The Murchison meteorite changed everything. When this 4.5 billion-year-old rock slammed into Australia in 1969, researchers found something impossible inside: amino acids. Not just any amino acids, but 74 different types, including many that don't exist naturally on Earth. These weren't contamination from our planet. The chemical signatures proved they formed in deep space. Think of amino acids as biological Lego blocks. Every protein in your body, from the hemoglobin carrying oxygen in your blood to the enzymes digesting your food, is built from these essential components. Finding them in a meteorite was like discovering a construction kit for life floating through the cosmos. But the Murchison meteorite wasn't alone. The Allende meteorite, which fell in Mexico, contained even more complex organic compounds. Scientists found nucleotide bases, the letters that spell out genetic codes in DNA and RNA. The Murray meteorite revealed sugar compounds that form the backbone of genetic material. The Carbon Hill meteorite contained lipids, the building blocks of cell membranes. Here's where things get really mind-bending. Some of these meteorites are older than our solar system. They formed in stellar nurseries billions of years before Earth existed, carrying their biological cargo across unimaginable distances and time spans. It's like receiving a message in a bottle from civilizations that lived and died before our sun was even born. Recent analysis has revealed something even more shocking. Some meteorites contain microscopic structures that look exactly like fossilized bacteria. These aren't random mineral formations. They have the distinct shapes, sizes, and internal organizations of living cells. The ALH 84001 meteorite, originally from Mars, sparked intense debate when NASA scientists claimed to have found evidence of ancient Martian life preserved in its rocky matrix. The implications are staggering. If meteorites regularly carried organic compounds throughout the early solar system, then the building blocks of life were being delivered to planets like a cosmic Amazon service. Earth, Mars, Venus, and the moons of Jupiter and Saturn were all receiving the same biological packages from deep space. This delivery system is still active today. Every time you see a meteor shower, you're watching potential life seeds rain down on our planet. The Perseid meteors, the Geminids, the Leonids, each streak of light represents organic compounds being deposited in our atmosphere. Some scientists now believe that Earth's early bombardment period, when asteroids and comets constantly pelted our planet, wasn't just a destructive force. It was the universe's way of planting a garden. Each impact delivered new genetic material, new biological possibilities, new evolutionary pathways. But if meteorites can carry life's building blocks across space, the next question becomes even more extraordinary. Can actual living organisms survive the journey? The answer lies in the most extreme creatures on our planet, organisms so tough they seem designed for interstellar travel. Earth sits in the most perfect spot imaginable, 93 million miles from a perfectly sized star. We call this the Goldilocks zone. Not too hot, not too cold, but just right for liquid water to exist. Move us 5% closer to the sun, and we become Venus, with surface temperatures hot enough to melt lead. Shift us 10% farther away, and we turn into Mars, a frozen desert where water exists only as ice and vapor. The mathematical precision is breathtaking. Our planet's orbit varies by less than 2% throughout the year. Our moon is exactly the right size to stabilize our axial tilt, preventing wild climate swings that would make complex life impossible. Our atmosphere is 78% nitrogen and 21% oxygen, a combination so chemically reactive it should have burned away billions of years ago. Yet somehow, this delicate balance has remained stable for billions of years. The odds of this happening by chance are so small that scientists struggle to express them mathematically. It's like flipping a coin and getting heads 10,000 times in a row. But here's what makes the Goldilocks problem even stranger. Earth wasn't always this perfect. 4 billion years ago, our sun was 25% dimmer than it is today. By all calculations, our planet should have been a frozen snowball, completely inhospitable to life. Scientists call this the faint young sun paradox. Yet somehow, during this supposedly impossible period, life not only existed but thrived. The geological record shows clear evidence of liquid oceans and complex microbial ecosystems. Something was keeping Earth warm when it should have been cold, maintaining conditions that allowed life to establish itself and spread. Traditional theories struggle to explain this. Some scientists suggest massive volcanic activity created a greenhouse effect. Others propose that early Earth had a much thicker atmosphere, but these explanations require a series of coincidences so improbable that they strain credibility. The Panspermia hypothesis offers a different perspective. What if Earth's perfect conditions aren't accidents, but engineering? What if advanced civilizations, facing the death of their own worlds, didn't just send out genetic material randomly? What if they deliberately chose target planets and modified them to be suitable for life? Consider the precision required. You'd need to adjust atmospheric composition, orbital mechanics, magnetic field strength, and planetary rotation. You'd need to account for stellar evolution, ensuring your chosen world remained habitable as its star aged and brightened. This level of terraforming would require knowledge and technology far beyond our current capabilities. Evidence for this kind of deliberate modification might be hidden in plain sight. Earth's unusually large moon, for instance, could be the result of an intentional collision designed to create tidal forces and stabilize our planet's rotation. Our magnetic field, generated by a liquid iron core, protects us from deadly solar radiation in a way that seems almost too convenient. Even our position in the galaxy appears optimized for life. We orbit in what astronomers call the galactic habitable zone, far enough from the galactic center to avoid destructive radiation, but close enough to benefit from the heavy elements created by dying stars. If Earth was deliberately prepared as a biological laboratory, then the next question becomes crucial. What kinds of organisms could actually survive the journey from their dying worlds to our carefully prepared one? Deep in the Atacama Desert, where it hasn't rained for over 400 years, scientists have found bacteria thriving inside rocks. These organisms, called endoliths, live in conditions that would kill almost any other life form on Earth. No water, no oxygen, no organic nutrients, and radiation levels that would be lethal to humans. Yet they not only survive but reproduce, creating entire ecosystems in what appears to be solid stone. This discovery led researchers to a startling question. If life can exist in Earth's most extreme environments, what about the ultimate extreme environment, outer space itself? The answer came from experiments aboard the International Space Station. Scientists exposed bacteria samples to the vacuum of space, where temperatures swing from 250 degrees below zero to 250 degrees above. No air pressure, no protection from cosmic radiation, no nutrients, no water. These conditions would kill a human being in seconds. The bacteria not only survived, but continued to reproduce. Deinococcus radiodurans, nicknamed Conan the bacterium, can withstand radiation doses 3,000 times higher than what would kill a human. It can survive in a complete vacuum for years. When its DNA gets shredded by cosmic rays, it simply rebuilds itself using internal repair mechanisms that scientists are still trying to understand. Tardigrades, microscopic animals that look like 8-legged bears, are even more impressive. They can shut down their metabolism completely, entering a state called cryptobiosis, where they're neither alive nor dead. In this state, they can survive temperatures of negative 458 degrees Fahrenheit, just a few degrees above absolute zero. They can handle pressure 6 times greater than the deepest ocean trenches. They can go without food or water for over 30 years. But here's the truly mind-bending part. When researchers exposed tardigrades to the vacuum of space for 10 days, 70% of them came back to life once they returned to normal conditions. They resumed eating, reproducing, and behaving as if nothing had happened. These aren't isolated examples. Scientists have found bacteria living in nuclear reactors, in pools of concentrated acid, in boiling hot springs, and in Antarctic ice that's been frozen for millions of years. Each discovery expands our understanding of life's incredible resilience and adaptability. If organisms this tough exist naturally on Earth, imagine what kinds of life forms might exist on planets with even harsher conditions. Worlds orbiting dying stars, planets with thick, toxic atmospheres, moons covered in radiation, bombarded ice. Life might not just survive in these environments, it might thrive. This changes everything about interstellar travel for living organisms. We used to think that life was fragile, requiring perfect conditions to survive even short journeys through space. Now we know that some Earth organisms could potentially survive trips lasting thousands or even millions of years. The implications are staggering. If bacteria and tardigrades can survive in space, they could be hitchhiking on asteroids and comets right now, traveling between star systems like biological stowaways. Every meteorite impact could be delivering not just organic compounds, but actual living passengers from distant worlds. But survival is only half the equation. For panspermia to work, these cosmic travelers would need to find suitable conditions for growth once they arrive. The question is whether Earth's fossil record shows any evidence of sudden biological arrivals that can't be explained by gradual evolution. Charles Darwin called it the most serious objection to his theory of evolution. The fossil record, which should show gradual transitions between species, instead reveals something far more puzzling. Complex life forms appear suddenly, fully formed, with no clear ancestral lineages. It's like finding modern smartphones in archaeological layers, without any evidence of earlier communication devices. Take the Cambrian explosion, 540 million years ago. For 3 billion years, Earth was populated almost exclusively by simple, single-celled organisms and basic multicellular forms. Then, in a geological instant spanning just 25 million years, virtually every major animal group appeared simultaneously in the fossil record. We're talking about creatures with complex eyes, sophisticated nervous systems, intricate shells, and advanced hunting strategies. Trilobites emerged with compound eyes containing over 15,000 individual lenses, more sophisticated than most modern insects. Anomalocaris, a 6-foot-long predator, appeared with flexible arms and complex mouthparts designed for processing prey. Where are their ancestors? Where are the transitional forms showing gradual development of these features? Traditional evolution predicts we should find millions of intermediate fossils, showing step-by-step progression from simple to complex. Instead, we find gaps so large that paleontologists have given them a name, the Cambrian Gap. But the Cambrian explosion isn't unique. The fossil record is filled with similar sudden appearances. Birds appear in the Jurassic period without clear reptilian ancestors, showing the gradual development of flight. Flowering plants emerge in the Cretaceous period without obvious precursors. Mammals diversify rapidly after the dinosaur extinction, filling ecological niches with stunning speed. Even more puzzling, these rapid appearances often coincide with asteroid impacts and cosmic bombardment periods. The Permian-Triassic extinction, the largest mass extinction in Earth's history, was followed by an explosion of new life forms. The asteroid impact that killed the dinosaurs led to mammalian diversification. Each catastrophic event seems to trigger waves of biological innovation. Modern genetics adds another layer to this mystery. When scientists analyze DNA sequences across different species, they find that many crucial genetic innovations appear suddenly in the evolutionary timeline. The genes responsible for complex eyes, for instance, are remarkably similar across vastly different animal groups, suggesting they didn't evolve independently, but were inherited from a common source. Some of the most important biological systems show this same pattern of sudden emergence. Photosynthesis, the process that converts sunlight into energy, appears fully formed in the fossil record, with no clear evolutionary pathway. The complex machinery required for DNA replication and protein synthesis emerges without obvious transitional stages. Traditional evolutionary theory explains these gaps as artifacts of poor fossil preservation. Soft-bodied organisms, they argue, simply don't fossilize well, creating an incomplete record. But this explanation struggles with the mathematical improbability of so many complex systems appearing simultaneously. The Panspermia hypothesis offers an alternative explanation. What if these sudden appearances represent biological colonization events? What if cosmic seeding doesn't just deliver simple organic compounds, but actual genetic blueprints from civilizations that had already solved these evolutionary challenges? If Earth has been receiving biological packages from space for billions of years, we might expect to find evidence of this delivery system still operating today. The key is knowing where to look and what cosmic environments might be manufacturing the building blocks of life right now. Between the stars, in regions so cold that atoms barely move, something extraordinary is happening. Vast clouds of cosmic dust, stretching across light years of space, are spontaneously creating complex organic molecules. These aren't random chemical reactions. Radio telescopes have detected over 200 different organic compounds floating in interstellar space, including many of the essential building blocks of life. The Eagle Nebula, 7,000 light-years away, contains enough organic material to seed millions of planets. The Orion Nebula produces alcohol molecules by the trillion, along with sugars, amino acids, and even more complex structures that scientists are still trying to identify. These stellar nurseries aren't just forming new stars and planets, they're manufacturing biological chemistry on an industrial scale. How is this possible in the vacuum of space? The secret lies in tiny dust grains, each smaller than cigarette smoke particles. These microscopic factories provide surfaces where atoms can meet and bond, protected from the harsh radiation that would normally tear molecules apart. Over millions of years, simple hydrogen and carbon atoms combine into increasingly complex structures. Think of each dust grain as a cosmic laboratory. Hydrogen atoms stick to the surface and gradually accumulate. When ultraviolet light from nearby stars hits these grains, it provides just enough energy to drive chemical reactions without destroying the products. The process is like slow cooking on a cosmic scale, where patience and precise conditions create molecular complexity that shouldn't exist in deep space. Recent observations from the James Webb Space Telescope have revealed something even more astonishing. Some interstellar clouds contain molecular structures so complex that they rival the chemistry found inside living cells. Long-chain hydrocarbons, ringed organic compounds, and even primitive membrane-like structures have been detected floating in regions where temperatures are 400 degrees below zero. These discoveries fundamentally change how we think about the distribution of life's building blocks. Instead of organic chemistry being rare and confined to planetary surfaces, we now know it's happening continuously throughout the galaxy. Every star-forming region is simultaneously a molecule-forming region, churning out biological materials that get incorporated into new solar systems. When these dust clouds eventually collapse to form stars and planets, they don't lose their organic inventory. Instead, all those complex molecules get concentrated into asteroids, comets, and planetary atmospheres. It's like the universe has been running a three-dimensional printing operation for billions of years, manufacturing biological components and distributing them to every corner of the galaxy. The scale is mind-boggling. Our Milky Way galaxy contains billions of these molecular factories, each one producing organic compounds equivalent to the mass of thousands of Earths. The rate of production means that every solar system, including ours, formed from material that was already rich in biological precursors. This cosmic chemistry set helps explain why organic compounds are found in meteorites from the earliest periods of solar system formation. It wasn't random chance that delivered these materials to Earth. It was an inevitable consequence of our cosmic environment, where the building blocks of life are as common as hydrogen and helium. But identifying organic compounds in space is just the beginning. The real question is whether we can find direct evidence of biological activity reaching Earth from these stellar laboratories. On July 20th, 1976, something unprecedented happened in the history of human exploration. The Viking 1 lander touched down on the Martian surface and immediately began conducting experiments designed to detect signs of life. What happened next launched a scientific controversy that continues to this day and potentially provided the first direct evidence of panspermia in action. The labeled release experiment was elegantly simple. The lander scooped up Martian soil and mixed it with a nutrient solution containing radioactive carbon. The logic was straightforward. If microorganisms existed in the soil, they would consume the nutrients and release radioactive carbon dioxide gas that sensitive detectors could measure. The results were immediate and dramatic. Within hours, the detectors registered a sharp spike in radioactive gas production. The pattern looked exactly like what you'd expect from biological metabolism. Living organisms were apparently eating the nutrients and breathing out radioactive carbon dioxide. Dr. Gilbert Levin, the experiment's designer, was convinced they had found life on Mars, but NASA remained skeptical. They ran additional tests, including control experiments, where soil samples were heated to temperatures that should kill any microorganisms. When heated samples showed no gas production, it seemed to confirm that something biological was responsible for the original results. However, other experiments on the same mission produced conflicting data. The gas chromatograph, designed to detect organic molecules, found no evidence of carbon-based compounds in the Martian soil. How could there be living organisms without organic chemistry to support them? For decades, the scientific consensus dismissed the Viking results as chemical rather than biological. Mars, they argued, was too cold, too dry, and too bombarded by radiation to support life. The gas production must have been caused by unknown chemical reactions in the alien soil, but this explanation never fully satisfied critics. The patterns detected by Viking were remarkably similar to biological signatures. The rapid initial response, followed by gradual decline, matched the behavior of Earth microorganisms introduced to new nutrient sources. Chemical reactions typically produce steady, predictable gas output, not the complex patterns Viking observed. Recent discoveries have forced scientists to reconsider these 40-year-old results. We now know that life on Earth exists in environments far more extreme than the Martian surface. Bacteria thrive in Antarctic ice, in highly acidic volcanic springs, and in environments with radiation levels that would be lethal to humans. If life can survive in these conditions on Earth, why not on Mars? More importantly, we've learned that organic compounds can be destroyed by the same radiation that might allow life to persist underground. The Viking landers only sampled the surface, where organic molecules would be rapidly broken down by ultraviolet radiation and cosmic rays. Any organic chemistry supporting Martian life would likely exist meters below the surface, beyond the reach of the Viking experiments. The implication is staggering. If the Viking experiments actually detected Martian life in 1976, it means we've had evidence of panspermia for nearly 50 years. Mars and Earth, both targets of the same cosmic bombardment, both recipients of organic compounds from interstellar space, might host related forms of life. The genetic evidence for this connection might be hidden in the most fundamental aspect of biology itself, the universal language that all Earth life uses to store and transmit information. Every living thing on Earth, from bacteria to blue whales, uses exactly the same genetic code. This universal language consists of just 4 chemical letters: A, T, G, and C. These letters combine into 3-letter words called codons, which tell cells how to build proteins. There are 64 possible three-letter combinations, but they code for only 20 amino acids. This means the genetic code contains built-in redundancy, like a language designed to survive transmission errors. The mathematical precision is extraordinary. The genetic code is optimized to minimize the damage from mutations. If cosmic radiation changes one letter in a genetic word, the redundancy ensures that the resulting protein is usually still functional. It's as if someone designed this system specifically to survive the harsh conditions of interstellar space. But here's what makes scientists pause. This level of optimization shouldn't exist if the genetic code evolved randomly on Earth. Computer simulations show that there are millions of possible genetic codes, most of which would work perfectly fine for life on a stable planet. Yet somehow, out of all these possibilities, life on Earth uses the one code that happens to be nearly optimal for surviving cosmic radiation. The probability of this happening by chance is roughly 1 in a million. It's like randomly generating a computer password and discovering it's been perfectly optimized for security by someone who understood advanced cryptography. Recent analysis of the genetic code has revealed even stranger patterns. The way DNA letters are arranged follows mathematical principles that weren't discovered by human scientists until the 20th century. The genetic code shows evidence of error-correcting mechanisms, redundancy systems, and information compression techniques that rival our most sophisticated computer programming. Some researchers have found what they call wow signals hidden in genetic sequences. These are mathematical patterns so precise and unlikely that they appear to be intentionally embedded messages. The Fibonacci sequence, prime numbers, and other mathematical constants seem to be woven into the fundamental structure of DNA itself. Consider the implications. If these patterns are intentional, then the genetic code isn't just a random product of Earth's evolution. It's a designed system created by intelligences that understood both biology and mathematics at levels we're only beginning to appreciate. The timing adds another layer of mystery. The universal genetic code appears to have been established very early in Earth's history, during the period when our planet was being heavily bombarded by asteroids and comets. This is exactly when we'd expect cosmic seeding to have the greatest impact on terrestrial biology. Even more intriguing, the genetic code shows signs of being compressed for efficient transmission. Like a digital file optimized for downloading, DNA contains the maximum amount of information in the minimum amount of space. This suggests it was designed for long-distance travel through space, where every bit of information has to survive radiation damage and thermal extremes. The universality of this code across all Earth life also supports panspermia. If life evolved independently from multiple seeding events, we might expect to see different genetic languages for different lineages. Instead, every organism uses the same system, suggesting a single source of information that was distributed globally. If the genetic code carries signatures of cosmic engineering, then we might expect to find similar evidence of extraterrestrial influence in the most unexpected places. Sometimes the most convincing evidence comes from the worlds we least expect to support life. Beneath 15 miles of solid ice, Europa hides an ocean twice as large as all of Earth's seas combined. Jupiter's fourth largest moon shouldn't exist in any form that supports life. At 500 million miles from the Sun, surface temperatures plunge to negative 260 degrees Fahrenheit. Solar radiation is 25 times weaker than what reaches Earth. By every conventional measure, Europa should be a lifeless ice ball, yet something extraordinary is happening beneath that frozen surface. Gravitational forces from Jupiter's massive pull flex Europa's rocky core, generating enough heat to maintain liquid water for billions of years. This subsurface ocean contains more water than all the rivers, lakes, and seas on our planet. And where there's liquid water, there might be life. Recent analysis of Europa's thin atmosphere has detected water vapor plumes erupting from the surface, shooting hundreds of miles into space. These geysers provide a direct window into the hidden ocean below. When the Hubble Space Telescope analyzed the chemical composition of these plumes, it found something remarkable. The water signature matches Earth's oceans almost perfectly. The similarity isn't just chemical. Isotope ratios, which act like fingerprints for different water sources, suggest that Europa's ocean and Earth's seas might share a common origin. This is difficult to explain if both worlds formed independently from local materials in their respective regions of the solar system. But Europa's connection to Earth might go far deeper than just water. The gravitational dynamics that keep Europa's ocean liquid also create powerful currents and chemical mixing that could support complex ecosystems. Volcanic vents on the ocean floor, similar to the hydrothermal systems that support life in Earth's deepest seas, might provide energy for biological processes. The most intriguing possibility is that Europa's ocean isn't just potentially habitable, it might be actively exporting life to other worlds. Every time those massive geysers erupt, they launch water, dissolved minerals, and potentially biological material into space. Some of this material escapes Europa's weak gravity and enters the broader solar system. Jupiter's powerful magnetic field can accelerate these particles to incredible speeds, sending them toward the inner solar system where Earth orbits. Over billions of years, this process could have delivered countless biological packages from Europa's hidden biosphere to our planet's surface. The timing is perfect for early Earth. During the late heavy bombardment period 4 billion years ago, the solar system was chaotic enough to facilitate massive material exchange between worlds. Asteroids and comets regularly carried material from the outer solar system to the inner planets. Europa's ocean, already mature and potentially teeming with life, could have been seeding Earth during this crucial period. This scenario explains several puzzles about early life on Earth: the rapid appearance of complex cellular mechanisms, the optimization of biochemical processes for surviving radiation, and the universal nature of the genetic code. All make sense if life arrived here, already tested and refined in Europa's ancient ocean. Modern space missions are actively testing this hypothesis. The upcoming Europa Clipper mission will analyze those water plumes in unprecedented detail, searching for organic compounds and potentially even direct signs of biological activity. If Europa is indeed connected to Earth's biological history, it raises a profound question about the nature of major evolutionary leaps in our planet's fossil record. 66 million years ago, a 6-mile-wide asteroid slammed into Earth with the force of 10 billion atomic bombs. The Chicxulub impact should have been the end of complex life on our planet. The explosion vaporized rock, created global tsunamis, and threw so much debris into the atmosphere that sunlight was blocked for months. 3/4 of all species went extinct, including the dinosaurs that had dominated Earth for 160 million years. But something unexpected happened in the aftermath of this cosmic catastrophe. Instead of a long, slow recovery, life exploded into forms more diverse and complex than ever before. Mammals, previously small creatures living in the shadows of dinosaurs, rapidly diversified into thousands of new species. Flowering plants flourished. New ecological niches were filled with stunning speed. This pattern isn't unique to the Chicxulub impact. Throughout Earth's history, major asteroid strikes have been followed by remarkable biological innovation. The Permian-Triassic extinction, triggered by massive volcanic activity and potentially cosmic impacts, was followed by the rise of archosaurs, the ancestors of dinosaurs and crocodiles. The late Ordovician extinction preceded an explosion of marine biodiversity. The correlation is so consistent that scientists have started mapping impact craters against the fossil record. What they've found challenges conventional thinking about evolution and extinction. Rather than just destroying life, cosmic impacts seem to introduce new biological possibilities. Consider the mechanics of what happens during a major impact. The explosion doesn't just kill organisms. It also creates conditions that could support the survival and growth of cosmic hitchhikers. The massive heat and pressure can create hydrothermal systems that persist for thousands of years. These environments, similar to the deep sea vents where life might have first emerged on Earth, could provide perfect nurseries for extraterrestrial microorganisms. The Vredefort impact structure in South Africa, 2 billion years old and 190 miles across, shows geological evidence of long-lived hydrothermal activity. Rock samples from the crater contain unusual mineral formations and chemical signatures that suggest biological processes were active for centuries after the impact. Even more intriguing, some impact sites show evidence of rapid organic compound formation in the immediate aftermath of collision. The Sudbury impact in Canada, 1.85 billion years old, contains organic carbon deposits that formed shortly after the impact event. These aren't just random chemical reactions. The molecular patterns suggest organized biological processes. Recent computer simulations reveal that asteroid impacts can actually protect biological material during atmospheric entry. While surface rocks get heated to thousands of degrees, the interior of large meteorites can remain relatively cool. Microorganisms embedded deep within incoming asteroids could survive the journey and find themselves in environments specifically created by the impact event. This creates a perfect biological delivery system. Cosmic impacts simultaneously destroy existing ecosystems, creating evolutionary opportunities, while delivering new genetic material that can exploit those opportunities. It's like clearing land and planting seeds in a single operation. The evidence suggests that Earth's evolutionary leaps aren't just products of random mutation and natural selection. They might be the result of periodic biological resupply missions from space, each one introducing new possibilities that reshape life on our planet. But if cosmic impacts regularly deliver living organisms to Earth, we might expect to find creatures that seem almost purpose-built for surviving the journey between worlds. Under a microscope, tardigrades look like tiny 8-legged bears wandering through a miniature landscape. These microscopic animals, smaller than a grain of salt, are found everywhere on Earth. They live in moss, lichen, soil, and water from the Arctic to the equator. But what makes tardigrades truly extraordinary isn't where they live, but what they can survive. When conditions become hostile, tardigrades enter a state called cryptobiosis, essentially becoming neither alive nor dead. Their metabolism drops to 0.01% of normal levels. They lose 99% of their body water and curl into an indestructible barrel called a ton. In this state, they can survive conditions that would obliterate almost any other life form on Earth. Temperature extremes that would instantly kill humans barely affect tardigrades. They've survived exposure to negative 458 degrees Fahrenheit, just a few degrees above absolute zero. They've also withstood temperatures of 300 degrees Fahrenheit. They can handle pressure 6 times greater than the deepest ocean trenches and radiation levels 6,000 times higher than what would be lethal to humans. Most remarkably, tardigrades are the only known animals that can survive the vacuum of space. When European Space Agency researchers exposed tardigrades to the harsh conditions outside the International Space Station for 10 days, 70% came back to life once returned to normal conditions. They had survived zero air pressure, cosmic radiation, and temperature swings that would destroy most known materials. This resilience seems almost purposefully engineered for interplanetary travel. Tardigrades can remain in cryptobiosis for decades, potentially long enough to journey between nearby star systems. Their ability to repair DNA damage from radiation is more sophisticated than anything human technology has achieved. When cosmic rays shred their genetic material, tardigrades simply rebuild it, using molecular mechanisms that scientists are still trying to understand. But here's what makes the tardigrade mystery even stranger. These survival abilities are completely unnecessary for their normal Earth lifestyle. Tardigrades typically live in mild, temperate environments where temperatures rarely exceed 100 degrees Fahrenheit and radiation levels are minimal. Why would evolution develop such extreme survival capabilities for organisms that rarely encounter extreme conditions? Traditional evolutionary theory suggests that traits develop in response to environmental pressures, yet tardigrades seem equipped for environments far more hostile than anything they encounter on Earth. It's like finding deep-sea diving equipment on animals that live in shallow ponds. Recent genetic analysis has revealed another puzzling aspect of tardigrade biology. Their DNA contains genetic sequences that don't match any other Earth organisms. Roughly 20% of tardigrade genes appear to have been acquired through horizontal gene transfer, but from unknown sources. This genetic material seems specifically designed for surviving extreme conditions. Some scientists now suggest that tardigrades might be living fossils from Earth's earliest biosphere, preserving survival traits from when our planet was regularly bombarded by asteroids and cosmic radiation. Others propose a more radical explanation. Tardigrades might be descendants of organisms that arrived on Earth from space, carrying genetic adaptations developed during interstellar journeys. The tardigrade mystery extends beyond individual survival to their role as potential carriers of other life forms. These microscopic travelers often host bacteria, viruses, and other microorganisms, creating biological cargo ships capable of transporting entire ecosystems through space. If tardigrades can survive interplanetary journeys, what about the cosmic delivery vehicles that might have brought them here in the first place? Every 76 years, Halley's Comet returns to our inner solar system, carrying a cargo more precious than any treasure on Earth. As this ancient wanderer approaches the Sun, solar radiation begins vaporizing its icy surface, creating the spectacular tale that has amazed humans for thousands of years. But hidden within that glowing display is something remarkable: complex, organic molecules, amino acids, and potentially even primitive life forms that have been preserved in deep freeze for billions of years. Comets are time capsules from the earliest days of our solar system. They formed in the Kuiper Belt and Oort cloud, regions so distant that they preserve original materials from when the planets first coalesced. Unlike asteroids, which have been heated and modified by their proximity to the Sun, comets retain pristine samples of primordial chemistry. When the European Space Agency's Rosetta mission finally caught up with Comet 67P in 2014, the discoveries revolutionized our understanding of these cosmic icebergs. The comet's nucleus contains 16 different amino acids, including glycine, the simplest building block of proteins. It harbored complex organic molecules like formamide, which plays crucial roles in the formation of nucleotide bases that make up DNA and RNA. But Rosetta found something even more extraordinary. The comet contained organic compounds organized into structures resembling primitive cell membranes. These weren't random chemical formations, but organized molecular arrangements that could potentially serve as containers for biological processes. The implications are staggering. If comets carry pre-biological structures, they're not just delivering raw materials for life. They're shipping partially assembled biological kits that could accelerate evolution on any planet they encounter. Consider the scale of this delivery system. Our solar system contains trillions of comets in the Oort cloud alone. Throughout cosmic history, gravitational perturbations have sent these icy messengers careening toward the inner planets. Earth has been bombarded by comets for over 4 billion years, each impact delivering tons of organic compounds directly to our planet's surface. The timing of major comet showers correlates remarkably with periods of rapid biological evolution on Earth. The late heavy bombardment, 4 billion years ago, coincided with the emergence of the first complex cells. Periodic comet storms throughout geological history align with evolutionary leaps that paleontologists struggle to explain through gradual processes alone. Modern analysis of comet dust collected by NASA's Stardust mission reveals that these cosmic wanderers carry isotope signatures matching life on Earth. The carbon, nitrogen, and sulfur found in comet samples show the same ratios found in terrestrial organisms, suggesting a shared origin or extensive material exchange. Even more intriguing, some comets show evidence of internal liquid water during their long journeys through space. Radioactive decay within comet nuclei can create warm zones where chemical evolution might continue during the millions of years between solar system visits. These wandering laboratories could be conducting biological experiments as they travel between star systems. If comets serve as biological cargo ships, then their role in panspermia extends far beyond our solar system. Gravitational interactions with passing stars regularly eject comets from planetary systems, sending them on interstellar journeys that could last millions of years. Some of these cosmic refugees eventually get captured by other star systems, delivering their biological payloads to distant worlds. This interstellar comet exchange might explain one of the most perplexing puzzles in modern astronomy: why we haven't found obvious signs of alien civilizations despite the universe being old enough for them to exist. In 1950, physicist Enrico Fermi asked a question that still haunts scientists today: where is everybody? The Fermi paradox highlights a troubling contradiction. Our galaxy contains over 400 billion stars, many with planetary systems that could support life. The universe is 13.8 billion years old, providing ample time for civilizations to arise and spread across the stars. Yet, despite decades of searching, we've found no conclusive evidence of alien intelligence. Traditional explanations for this silence are deeply unsettling. Perhaps alien civilizations destroy themselves before achieving interstellar travel. Maybe intelligence is so rare that we're truly alone in a vast, empty cosmos. Some scientists propose that hostile alien powers deliberately suppress the emergence of new civilizations, maintaining galactic silence through force. But the panspermia hypothesis suggests a radically different explanation. We're not looking for aliens because we are the aliens. Consider this possibility. Billions of years ago, when our galaxy was young and chaotic, advanced civilizations faced the inevitable death of their star systems. Rather than accepting extinction, they developed the ultimate survival strategy: cosmic reproduction. They encoded their biological heritage into microscopic passengers that could survive interstellar journeys lasting millions of years. These ancient civilizations didn't just send random genetic material into space. They carefully designed self-replicating systems optimized for long-distance travel and rapid colonization of suitable worlds. The universal genetic code, with its sophisticated error correction and radiation resistance, wasn't the product of random evolution. It was engineered by intelligences that understood both biology and cosmic conditions at levels we're only beginning to appreciate. From this perspective, Earth wasn't randomly chosen for seeding. It was deliberately prepared and monitored as part of a galaxy-spanning project to preserve and spread intelligence throughout the cosmos. The mathematical precision of our planet's orbit, the convenient presence of a large stabilizing moon, and the perfect atmospheric composition for complex life weren't cosmic accidents. They were the result of careful planetary engineering. This scenario explains the Fermi paradox perfectly. We don't detect alien civilizations because they're not separate from us. We are their descendants, carrying their genetic legacy and slowly redeveloping their technological capabilities. Every human being is a walking monument to civilizations that existed billions of years before our solar system formed. The silence we detect isn't emptiness, but success. The ancient seeding project worked so well that its products, including us, eventually develop their own technology and begin searching for the very civilizations that created them. We're looking for our ancestors without realizing that we carry their genetic signatures in every cell of our bodies. Recent advances in genetic analysis might be revealing traces of this heritage. The mathematical patterns hidden in DNA, the unexpected optimization of biological systems for surviving cosmic radiation, and the universal nature of genetic codes all point toward purposeful design rather than random evolution. If this interpretation is correct, then humanity's role in the cosmos is far more significant than we've imagined. We're not just random products of terrestrial evolution, but heirs to a galactic civilization that sacrificed itself to ensure that intelligence would continue throughout the universe. But if ancient civilizations deliberately seeded planets like Earth, the next question becomes, whether this process involved direct intervention rather than just passive genetic distribution. In 1973, Francis Crick made a startling proposal that shocked the scientific community. Crick, who had won the Nobel Prize for discovering the structure of DNA, suggested that life on Earth was too complex to have evolved naturally in the time available. Instead, he proposed that advanced extraterrestrial civilizations deliberately sent genetic material to our planet in unmanned spacecraft, specifically designed to seed suitable worlds with life. This wasn't random speculation from a fringe theorist. Crick had spent decades studying the intricate machinery of cellular biology and understood better than almost anyone how impossibly complex even the simplest living systems really are. His directed panspermia theory emerged from cold mathematical analysis of what evolution could and couldn't accomplish in Earth's brief history. The numbers that troubled Crick are even more troubling today. A single bacterial cell contains roughly 4 million precisely arranged molecular parts. The probability of these components spontaneously organizing into a functioning organism is so small that mathematicians struggle to express it. It's like a tornado sweeping through a junkyard and accidentally assembling a working jumbo jet, except the odds are infinitely worse. But directed panspermia solves this probability problem elegantly. Instead of requiring life to emerge from scratch on every planet, it suggests that intelligence, once achieved, takes responsibility for spreading itself throughout the galaxy. Advanced civilizations don't just survive by traveling to new worlds themselves. They ensure their biological legacy continues by deliberately seeding potentially habitable planets. Modern technology makes Crick's scenario increasingly plausible. We now understand how to design microscopic machines capable of self-replication and error correction. We know how to encode vast amounts of information in DNA sequences. We've identified the genetic modifications that would allow organisms to survive extended space travel. If we can envision these technologies after just decades of molecular biology research, imagine what civilizations with millions of years of development could achieve. The directed panspermia scenario explains several puzzles about Earth's biological history. The universal genetic code used by every living thing on our planet makes perfect sense as a designed system optimized for reliability and error correction. The rapid appearance of complex cellular machinery in the fossil record becomes logical if that machinery was engineered rather than evolved. Recent discoveries support the possibility that such seeding missions might have actually occurred. The Oumuamua object, which passed through our solar system in 2017, exhibited characteristics that some scientists interpreted as artificial rather than natural. Its unusual elongated shape, unexpected acceleration, and trajectory suggested it might have been a probe from another star system. If directed panspermia is real, then Earth's entire biosphere represents the successful execution of an ancient terraforming project. Every forest, every ocean ecosystem, every creature that has ever lived carries the genetic legacy of civilizations that died billions of years before our sun ignited. This perspective fundamentally changes how we think about our relationship with the cosmos. We're not just random accidents in an indifferent universe, but the intended outcomes of the greatest engineering project ever conceived. But for such an ambitious seeding project to succeed, it would require more than just sending genetic material into space. It would need to actively modify planetary environments to support the life forms being introduced. 2.5 billion years ago, Earth's atmosphere underwent the most dramatic transformation in our planet's history. For over 1 billion years, our world had been shrouded in a toxic atmosphere of methane, ammonia, and carbon dioxide. Oxygen, the gas that now sustains complex life, was virtually non-existent. Any organism that encountered free oxygen would have been poisoned by its reactive chemistry. Earth was essentially a giant chemical factory, running processes that would be lethal to every animal alive today. Then, in what geologists call the Great Oxygenation Event, everything changed. Oxygen levels skyrocketed from nearly 0 to 21% of the atmosphere in what amounts to a geological instant. This wasn't a gradual shift spanning hundreds of millions of years. It happened so rapidly that it triggered the first mass extinction in Earth's history, as organisms adapted to the old atmosphere were wiped out by the new oxygen-rich environment. Traditional science attributes this transformation to cyanobacteria, primitive organisms that developed photosynthesis and began converting carbon dioxide into oxygen. But the mathematics of this explanation creates serious problems. For cyanobacteria to produce the observed atmospheric changes, they would need to multiply at rates that exceed anything observed in modern ecosystems, while simultaneously developing biochemical processes more sophisticated than most modern organisms possess. The speed of the transformation is particularly puzzling. Computer models suggest that natural photosynthesis should have taken over 10 billion years to create our current atmosphere. Yet the Great Oxygenation Event accomplished this in less than 500 million years. It's like expecting a wood-burning stove to heat up a warehouse in minutes rather than hours. But there's another explanation that makes the timeline work perfectly. What if the Great Oxygenation Event wasn't a natural evolutionary development, but a deliberate terraforming operation? What if the cyanobacteria responsible for this atmospheric revolution were introduced as part of a cosmic seeding project, specifically designed to prepare Earth for complex life? Evidence for this interpretation lies in the biochemical sophistication of photosynthesis itself. This process requires two separate photosystems working in perfect coordination, along with complex molecular machinery for capturing light energy and converting it into chemical energy. The probability of all these components evolving simultaneously by random chance approaches mathematical impossibility. Yet photosynthesis appears fully formed in the earliest cyanobacteria fossils, with no evidence of the gradual development you'd expect from natural evolution. It's like finding modern computer chips in archaeological layers from the Stone Age. The timing of the Great Oxygenation Event also correlates with increased bombardment of Earth by asteroids and comets. Geological evidence shows unusual levels of cosmic material reaching our planet during this crucial period, suggesting that whatever triggered the atmospheric transformation arrived from space. Recent analysis of ancient rock formations has revealed another startling pattern. The isotope signatures in rocks from the Great Oxygenation Event don't match what we'd expect from purely terrestrial biological processes. Instead, they show characteristics that suggest involvement of organisms with metabolic pathways unknown in modern Earth life. If the Great Oxygenation Event was indeed an engineered transformation, then the organisms responsible for this planetary renovation might still exist on Earth today, hidden in the most extreme environments, where only the toughest life forms can survive. In the scalding hot springs of Yellowstone National Park, where temperatures reach 212 degrees Fahrenheit, something impossible thrives. Thermus aquaticus, a bacteria that lives in near boiling water, shouldn't exist according to traditional biology. Proteins denature at high temperatures, DNA breaks apart, and cellular membranes melt. Yet these remarkable organisms not only survive but flourish in conditions that would instantly kill most Earth life. The discovery of extremophiles has revolutionized our understanding of life's boundaries. Scientists have found organisms thriving in environments that mirror the harsh conditions of space and alien worlds. Acidophiles live in sulfuric acid concentrations that would dissolve metal. Psychrophiles remain active in Antarctic ice at temperatures of -40 degrees Fahrenheit. Piezophiles survive crushing pressures 11 times greater than the deepest ocean trenches. But the most remarkable extremophiles are the ones that seem specifically adapted for space travel. Deinococcus radiodurans can survive radiation doses 1,000 times higher than would be lethal to humans. When its DNA gets completely shredded by cosmic rays, it simply rebuilds its genetic code from fragments using repair mechanisms that remain mysterious to modern science. These repair capabilities go far beyond what natural selection on Earth would require. Our planet's surface receives minimal cosmic radiation compared to the levels encountered in deep space. Why would organisms evolve such sophisticated radiation resistance unless they needed it for environments far more hostile than Earth? Recent genetic analysis has revealed something even more extraordinary about extremophiles. Many of their survival traits are encoded by genes that don't match anything else in the terrestrial evolutionary tree. These organisms appear to have acquired their extreme survival capabilities through horizontal gene transfer from unknown sources. Horizontal gene transfer is biology's way of sharing useful traits between unrelated species. Bacteria regularly exchange genetic material, allowing rapid adaptation to new environments. But extremophiles show evidence of having received genetic packages that are far too sophisticated and coordinated to have originated from other Earth organisms. Consider Caldalcalibacillus thermorum, a bacteria that thrives in hot alkaline springs. Its genome contains genetic circuits for surviving conditions found on Jupiter's moon Io, where volcanic activity creates environments similar to its preferred habitat. Yet this bacteria evolved on Earth, where such extreme volcanic conditions are rare and localized. The most intriguing extremophiles are methanogens, organisms that produce methane as a waste product of their metabolism. These ancient bacteria can survive in environments completely devoid of oxygen, using hydrogen and carbon dioxide to generate energy. Their metabolic processes are so alien to normal Earth life that some scientists consider them living fossils from a different kind of biosphere. Methanogens are found everywhere on Earth where conditions are suitable, but they're also exactly the kind of organisms you'd expect to find on Mars, Europa, and other worlds in our solar system. Their metabolism doesn't just tolerate the conditions on these alien worlds, it's perfectly adapted for them. If these extremophiles are descendants of cosmic travelers, then they might represent the advance guard of a much larger biological invasion. Their role wouldn't just be survival, but preparation of Earth's environment for more complex life forms that would follow. The evidence for this kind of biological staging might be written in the most dramatic chapter of Earth's evolutionary history: the sudden appearance of complex animal life in what scientists call the Cambrian explosion. 540 million years ago, Earth's oceans suddenly filled with creatures that looked like they came from another planet. For 3 billion years, life on our world had been simple and small. Single-celled bacteria, primitive algae, and basic multicellular organisms dominated the seas. Then, in a geological instant spanning just 25 million years, virtually every major animal group appeared simultaneously in the fossil record, with fully developed body plans and complex anatomical features. Trilobites emerged with compound eyes, containing 15,000 individual lenses, more sophisticated than most modern insects. Anomalocaris, a 6-foot predator, appeared with flexible arms and complex feeding mechanisms. Opabinia sported 5 eyes and a bizarre trunk tipped with a grasping claw. These weren't simple organisms gradually developing new features. They were biological masterpieces, appearing without obvious evolutionary ancestors. The mathematical improbability is staggering. Modern evolutionary biologist Stephen Jay Gould calculated that the innovations seen in the Cambrian period would require genetic changes occurring at rates millions of times faster than anything observed in recorded history. It's like watching the entire automotive industry evolve, from horse-drawn carriages to Formula One race cars in a single decade. Traditional explanations struggle with this timeline. Some scientists propose that rising oxygen levels triggered rapid evolution, but this doesn't explain why complex body plans appeared simultaneously across unrelated lineages. Others suggest that predator-prey arms races accelerated development, but this requires pre-existing predators that don't appear in the fossil record. The Panspermia hypothesis offers a radically different explanation. What if the Cambrian explosion represents the large-scale introduction of genetic templates from advanced biospheres elsewhere in the galaxy? What if Earth's primitive organisms suddenly gained access to biological blueprints that had been tested and refined on countless other worlds? Evidence for this interpretation lies in the sophistication of Cambrian innovations. Complex eyes, nervous systems, digestive organs, and circulatory systems all appear fully formed with no evidence of gradual development. These biological systems are so interdependent that they could only function as complete packages, not as gradually assembled components. Recent genetic analysis has revealed another troubling aspect of the Cambrian explosion. Many of the genes responsible for complex animal development don't appear gradually in the evolutionary timeline. Instead, they show up suddenly during the Cambrian period, often in multiple unrelated lineages simultaneously. The Hox genes, which control body plan development in all complex animals, exemplify this pattern. These genetic control systems are so sophisticated that they regulate the formation of heads, limbs, organs, and body segments with mathematical precision. Yet they appear in the fossil record without obvious precursors, as if they were introduced from an external source. Even more puzzling, many Cambrian organisms possess biological innovations that wouldn't be reinvented by evolution for hundreds of millions of years. Some early arthropods had joint mechanisms more sophisticated than modern insects. Certain Cambrian mollusks developed optical systems that weren't seen again until the development of modern cephalopods. If the Cambrian explosion represents cosmic genetic engineering, then the delivery system for these biological upgrades might still be operating today. The question is whether we can identify the microscopic vehicles that continue to transport genetic material between different forms of life on our planet. Viruses are the most abundant life forms on Earth, outnumbering all other organisms combined by a factor of 10 to 1. These microscopic entities exist in a strange twilight zone between living and non-living matter. They can't reproduce without hijacking the cellular machinery of other organisms, yet they carry genetic information and evolve like any biological system. What makes viruses truly extraordinary is their role as genetic delivery vehicles, constantly transferring DNA sequences between unrelated species in ways that traditional evolution can't explain. Recent discoveries have revealed that viruses are far more than simple disease-causing agents. They're sophisticated genetic engineers, capable of inserting new traits into host organisms with surgical precision. Retroviruses can permanently integrate their genetic material into host DNA, creating heritable changes that get passed to future generations. Some viruses carry genetic modules that enhance their host's survival capabilities rather than harming them. The scale of viral genetic transfer is mind-boggling. Scientists estimate that 8% of the human genome consists of viral DNA sequences that were incorporated by our ancestors millions of years ago. These aren't random insertions, but carefully integrated genetic programs that often provide beneficial functions. Some viral sequences regulate immune system responses, others control embryonic development. Many appear to enhance our ability to adapt to environmental changes. But here's what makes viral genetic transfer particularly intriguing from a panspermia perspective. Viruses can survive conditions that would destroy most other life forms. They've been found in the upper atmosphere, in deep ocean sediments, and embedded in ice that's been frozen for hundreds of thousands of years. Their simple structure and lack of metabolism make them nearly indestructible during long-term storage. If viruses can survive extended periods in hostile environments, they could potentially survive interplanetary journeys. Cosmic radiation that would shred complex cellular machinery might leave viral genetic packages intact. Extreme temperatures and vacuum conditions that would kill bacteria might preserve viral DNA indefinitely. Recent analysis of viral diversity has revealed patterns that are difficult to explain through terrestrial evolution alone. Many viruses carry genetic sequences that don't match any known Earth organisms. These orphan genes appear to encode functions that are useful for survival in extreme environments, yet they're found in viruses that infect organisms living in moderate conditions. Some viruses show evidence of carrying genetic material between vastly different types of organisms in ways that shouldn't be possible through normal biological processes. Marine viruses transfer genes between bacteria and algae. Atmospheric viruses move genetic material between plants and fungi. These transfer events happen too frequently and too precisely to be random accidents. The most intriguing possibility is that viruses might serve as biological memory banks, preserving genetic information from extinct civilizations and gradually releasing it into new host organisms. If ancient spacefaring species encoded their biological knowledge into viral delivery systems, these genetic time capsules could still be operating on Earth today. Evidence for this kind of cosmic genetic archaeology might be found in the most unexpected places. Sometimes, the most compelling proof of extraterrestrial influence literally falls from the sky in forms that mainstream science has been reluctant to acknowledge. On July 25, 2001, the skies over Kerala, India began raining blood. For 2 months, crimson droplets fell from clear skies across a 100-square-mile area. Local residents reported that the rain left red stains on clothing and buildings that were nearly impossible to remove. Government officials initially dismissed the phenomenon as dust from distant deserts, but laboratory analysis revealed something far more extraordinary. The red coloration came from microscopic cells, unlike anything in Earth's known biology. These organisms were roughly 4 micrometers in diameter, about half the size of typical bacteria, with thick cell walls and unusual internal structures. When scientists tried to culture them using standard biological techniques, the cells refused to grow in any known nutrient medium. But here's where the story becomes truly remarkable. The red rain cells showed characteristics that seemed specifically adapted for surviving in space. They could withstand extreme temperatures, intense radiation, and complete dehydration. When exposed to conditions that would kill most Earth bacteria, the mysterious red cells remained intact and apparently viable. Dr. Godfrey Louis, a physicist at Mahatma Gandhi University, spent years analyzing these cells and reached a startling conclusion. The organisms weren't terrestrial. Their cellular structure, chemical composition, and survival characteristics all suggested an extraterrestrial origin. Most significantly, they contained no DNA or RNA, the genetic molecules that define all known Earth life. Instead of conventional genetic material, the red rain cells appeared to use an entirely different information storage system. They reproduced by a process that resembled budding more than cellular division, and they could apparently survive indefinitely without nutrients or water. It was as if someone had designed organisms specifically for interplanetary travel and long-term storage in hostile environments. The timing of the red rain events added another layer of mystery. Many incidents occurred shortly after meteor showers or unusual atmospheric phenomena. Witnesses reported seeing bright flashes in the sky before the red droplets began falling. Some scientists suggested that the cells were being delivered by micrometeorites too small to be detected by conventional monitoring systems. Kerala wasn't an isolated case. Similar red rain events have been reported throughout history and around the world. Ancient texts describe blood rain falling during significant historical events. Modern incidents in Colombia, Austria, and Sri Lanka have yielded microscopic organisms with characteristics remarkably similar to the Kerala cells. Chemical analysis of these mysterious organisms has revealed isotope ratios that don't match terrestrial biology. The carbon signatures, in particular, suggest formation in environments with different stellar conditions than our solar system. These aren't just unusual Earth organisms. They appear to be genuine alien life forms. If the red rain cells are extraterrestrial visitors, they represent direct evidence that space-borne life regularly reaches Earth's surface. Each rainfall could be delivering millions of cosmic organisms, potentially introducing new genetic material into our planet's biosphere. The implications are staggering. We might be witnessing active panspermia in real time. But for cosmic seeding to work as a galactic strategy, it would need to operate with mathematical precision rather than random chance. The numbers behind panspermia reveal why ancient civilizations might have chosen this approach as their ultimate survival strategy. Imagine playing cosmic roulette, where the stakes are the survival of intelligence itself across the entire galaxy. Traditional approaches to interstellar colonization require massive resources and enormous risks. Building starships capable of carrying living passengers between star systems demands technologies and energy sources that stretch even advanced civilizations to their limits. The journey times span thousands of years, during which equipment fails, resources are depleted, and colonists face psychological pressures that could destroy entire missions. But panspermia changes the mathematical equation completely. Instead of sending a few expensive, fragile spacecraft to carefully chosen destinations, civilizations can launch billions of microscopic survival packages toward every potentially suitable planet in their galactic neighborhood. The cost per mission drops to nearly zero, while the probability of success skyrockets. Consider the numbers. Our galaxy contains over 400 billion stars, many with planetary systems that could support life. A single dying civilization could potentially seed thousands of nearby star systems using technology no more advanced than our current genetic engineering capabilities. Each seeding package would be designed to survive interstellar journeys, lasting millions of years while carrying complete biological blueprints for reconstructing complex life. The mathematics of survival probability become overwhelming in favor of the seeding strategy. If a civilization sends just 100 seeding packages to each of 1,000 nearby star systems, they've launched 100,000 biological time capsules into space. Even if 99% of these missions fail due to radiation damage, impact destruction, or unsuitable planetary conditions, 1,000 seeding operations still succeed. Each successful seeding event doesn't just create life on a new world. It establishes a biological beachhead that can eventually redevelop intelligence and technology. Given enough time, these new civilizations reach the point where they can launch their own seeding missions, creating an exponential expansion of intelligence throughout the galaxy. The timescales involved work in favor of this strategy. While conventional space travel requires civilizations to remain stable for thousands of years during transit, panspermia operates on geological timeframes where even unstable civilizations can achieve galactic impact. A society might collapse shortly after launching its seeding packages, but those biological messengers continue their journeys for millions of years. Recent computer simulations of galactic panspermia show that even conservative seeding rates could populate the entire Milky Way with life within 2 billion years. Given that our galaxy is over 13 billion years old, there's been more than enough time for multiple waves of cosmic seeding to occur. The redundancy built into this system makes extinction nearly impossible. Unlike conventional colonization, where the destruction of individual colonies threatens the entire expansion effort, panspermia creates thousands of independent backup copies of civilization. Even catastrophic events that sterilize entire star systems can't prevent the overall success of the seeding strategy. From this perspective, the absence of obvious alien civilizations in our galaxy isn't evidence that we're alone. It's evidence that panspermia worked so well that intelligence has been quietly spreading throughout the cosmos for billions of years, creating new forms of life that eventually develop their own technologies and continue the seeding process. But if galactic civilizations have been communicating through biological rather than technological means, we might need to reconsider what kinds of signals we should be looking for in our search for extraterrestrial intelligence. On August 15, 1977, astronomer Jerry Eman detected a radio signal so powerful and precisely focused that he wrote, "Wow," in the margin of the computer printout. The signal lasted exactly 72 seconds and came from the direction of the constellation Sagittarius. Its frequency was 1420 MHz, precisely the wavelength that hydrogen atoms emit when they change energy states. This wasn't random cosmic noise. It was a narrowband transmission that required technological sophistication to generate. Despite decades of searching, the WOW signal has never been detected again. Traditional SETI researchers assume it was a one-time transmission from an alien civilization, but the lack of repetition has puzzled scientists for nearly 50 years. Why would an advanced civilization send just one signal and then fall silent forever? The Panspermia hypothesis suggests a radically different interpretation. What if the WOW signal wasn't a technological transmission at all, but a biological one? What if it was the electromagnetic signature of a massive cosmic seeding event, where billions of genetic packages were being launched toward our solar system? Recent research has revealed that biological processes can generate electromagnetic emissions under specific conditions. When DNA replicates, it produces measurable electromagnetic fields. When cells divide rapidly, they create electromagnetic pulses that can be detected with sensitive equipment. Large-scale biological events involving trillions of organisms simultaneously could potentially generate radio signals powerful enough to be detected across interstellar distances. If ancient civilizations encoded genetic information into electromagnetic patterns, they could transmit biological blueprints at the speed of light rather than waiting millions of years for physical delivery. These transmissions wouldn't be trying to establish communication in the traditional sense. They would be broadcasting genetic instructions that could be received and implemented by any civilization with sufficient technological capability. The mathematics of this approach are compelling. A single electromagnetic transmission, lasting just minutes, could carry more genetic information than billions of physical seeding packages. The signal could be precisely targeted at star systems showing signs of developing intelligence, allowing advanced civilizations to accelerate the biological development of younger worlds. This perspective reframes the entire SETI enterprise. Instead of listening for messages intended for communication, we might need to search for transmissions designed for genetic engineering. These signals wouldn't repeat because they're not conversations but biological deliveries. Once the genetic package is transmitted, the mission is complete. Modern analysis of the WOW signal's characteristics supports this interpretation. The signal's duration, frequency stability, and power output are all consistent with what you'd expect from a sophisticated genetic transmission system. The precise hydrogen frequency could serve as a universal beacon, alerting intelligent civilizations that biological information is being broadcast. If this theory is correct, then we might have missed hundreds of similar transmissions simply because we weren't looking for the right kind of signal. Genetic broadcasts would appear as brief, powerful emissions, rather than the continuous signals that traditional SETI searches are designed to detect. The implications are staggering. Earth's biosphere might not just be the product of ancient physical seeding, but ongoing electromagnetic genetic programming from advanced civilizations throughout the galaxy. But biological transmissions from space might leave more than just electromagnetic signatures. They could create physical traces in Earth's geological record that reveal the true extent of cosmic influence on our planet's development. Deep beneath the Australian outback lies a rock formation that shouldn't exist on any planet, formed through natural processes. The Pilbara Craton contains mineral structures dating back 3.5 billion years that show characteristics typically associated with biological activity. These aren't simple, organic compounds, but complex crystalline formations that appear to have been shaped by living organisms using techniques that exceed anything in modern biology. Geologists studying these ancient formations have found microscopic tubes, spheres, and filaments embedded in rock that formed when Earth was still a hostile, radiation-bombarded world. The precision of these structures suggests they were created by organisms with sophisticated tools for manipulating matter at the molecular level. It's like finding evidence of nanotechnology in rocks that predate the evolution of complex cells by billions of years. Similar anomalous formations have been discovered in geological sites around the world. The Barberton Greenstone Belt in South Africa contains mineral deposits with isotope ratios that don't match any known terrestrial processes. The Isua supracrustal belt in Greenland shows evidence of biological carbon fixation occurring at temperatures and pressures that should have prevented any known form of life from surviving. But these ancient biological signatures aren't limited to obvious fossil sites. Some of Earth's most fundamental geological features show characteristics that suggest artificial modification rather than natural formation. The distribution of rare earth elements in certain rock layers follows patterns that are mathematically too precise to be random. Some mineral deposits show organization that resembles technological rather than geological processes. Recent analysis of impact crater formations has revealed another puzzling pattern. Many ancient impact sites show unusual mineral compositions that suggest the incoming objects weren't typical asteroids, but contained materials with biological origins. Chemical signatures in these craters indicate the presence of complex organic compounds that formed under conditions unlike anything in our solar system. The Sudbury impact structure in Canada, formed 1.85 billion years ago, contains mineral deposits that show evidence of rapid biological colonization immediately after the impact event. The speed of this colonization exceeds anything observed in modern impact sites, suggesting that the incoming object delivered organisms specifically adapted for exploiting post-impact environments. Even more intriguing are geological formations that appear to have been deliberately constructed rather than naturally formed. The Richat structure in Mauritania, often called the Eye of the Sahara, shows concentric ring patterns with mathematical precision that's difficult to explain through conventional geological processes. Some researchers suggest it might be the remains of an ancient artificial structure built by intelligences that understood planetary engineering on a massive scale. Underground cave systems in various parts of the world contain mineral formations that seem to have been shaped by unknown biological processes. These structures show levels of organization and complexity that exceed what modern organisms can achieve. The caves themselves often follow geometric patterns that suggest purposeful excavation rather than natural water erosion. If these geological anomalies represent evidence of ancient biological engineering, then Earth's entire geological history might need to be reinterpreted. What we consider natural formations could actually be the remains of cosmic civilizations that modified our planet billions of years before complex life evolved. The ultimate evidence for this reinterpretation might lie in the most fundamental aspect of biology itself, the remarkable uniformity of genetic systems across all Earth life. Every organism on Earth, from the tiniest bacteria to the largest whales, speaks exactly the same genetic language. This universal code consists of 64 three-letter words made from just 4 chemical letters, A, T, G, and C. These genetic words tell cells how to build the 20 amino acids that form the basis of all proteins. What makes this universality extraordinary is that there are millions of possible genetic codes that would work perfectly well for life, yet every Earth organism uses precisely the same one. The mathematical probability of this happening by random chance is essentially zero. It's like discovering that every human culture independently developed identical languages with the same vocabulary, grammar, and syntax. The only logical explanation is that all Earth life inherited this genetic language from a common source. But the universal genetic code shows characteristics that are even more remarkable than its uniformity. Computer analysis reveals that this particular code is optimized in ways that seem almost impossibly sophisticated. It contains built-in error correction mechanisms that minimize the damage from mutations. It's organized to maximize information storage while maintaining stability during replication. When scientists run computer simulations to test alternative genetic codes, they find that random variations almost always perform worse than the system used by Earth life. the probability of accidentally discovering such an optimized code through random evolution is roughly 1 in a million. It's like randomly typing on a keyboard and accidentally creating a computer program that's more efficient than anything human programmers have designed. Recent analysis has uncovered even stranger patterns hidden within the genetic code. Some researchers have found mathematical sequences embedded in the arrangement of genetic letters that correspond to fundamental constants in physics and mathematics. The Fibonacci sequence, prime numbers, and even pi appear to be encoded in the structure of DNA itself. These mathematical signatures suggest that the genetic code isn't just a biological accident, but a designed information system created by intelligences that understood both mathematics and biology at levels we're only beginning to appreciate. It's as if someone embedded a cosmic signature in the fundamental operating system of life itself. The universality of this code also explains why horizontal gene transfer works so seamlessly across different species. Viruses can move genetic material between bacteria, plants, and animals because all Earth organisms use the same genetic language. This compatibility wouldn't exist if different lineages had evolved independent genetic systems. If the universal genetic code represents the product of cosmic engineering, then its optimization for surviving radiation damage makes perfect sense. Space-traveling genetic material would need to survive cosmic rays, ultraviolet radiation, and high-energy particles during interstellar journeys. The error correction and redundancy built into Earth's genetic system seems specifically designed for these challenges. The code's mathematical elegance also suggests it was created for long-term preservation and transmission. Like a digital file compressed for efficient storage and download, the genetic code contains maximum information in minimum space, while maintaining perfect fidelity across billions of reproduction cycles. This level of sophisticated design implies that whoever created the universal genetic code understood biological and mathematical principles that human science is only now discovering. We might be using genetic technology designed by civilizations that existed billions of years before our solar system formed. But the most compelling evidence for ongoing cosmic influence on Earth's biology might come from current space missions that are discovering signs of life throughout our solar system. The Perseverance rover is currently drilling into Martian rocks that contain organic compounds identical to those found in Earth's earliest fossils. On February 18, 2021, this nuclear-powered laboratory landed in Jezero crater and immediately began finding evidence that Mars once hosted conditions suitable for life. But the discoveries go far beyond just ancient water flows and suitable chemistry. Perseverance has detected complex organic molecules in rock samples that show characteristics of biological rather than geological formation. These aren't simple carbon compounds that could form through random chemical processes. They're sophisticated molecular structures that, on Earth, are produced exclusively by living organisms. The isotope ratios, molecular arrangements, and chemical patterns all point toward biological activity that occurred when Mars was warmer and wetter billions of years ago. Even more remarkable, some of these organic compounds show signs of recent formation. Martian soil samples contain molecules that appear to be actively changing, suggesting that biological processes might still be operating in Mars's subsurface environment. If life exists on Mars today, it's probably hidden meters below the surface, protected from radiation and extreme temperatures. The implications for panspermia are profound. Mars and Earth exchange material constantly through asteroid impacts that launch rocks between the two planets. If both worlds harbor related forms of life, it supports the idea that biological material regularly travels throughout our solar system. Meanwhile, the James Webb Space Telescope has been detecting organic compounds in the atmospheres of distant planets. These discoveries reveal that the building blocks of life are common throughout the galaxy, not rare accidents confined to our solar system. Complex molecules that were once thought to be products of terrestrial biology are now being found around stars hundreds of light years away. The Europa Clipper mission, scheduled to arrive at Jupiter's moon in 2030, will search for signs of life in the subsurface ocean that contains more water than all of Earth's seas combined. Preliminary analysis of water vapor plumes erupting from Europa's surface has already detected organic compounds and possible amino acids, but perhaps the most intriguing discoveries are coming from Earth's own upper atmosphere. High-altitude research flights have detected microorganisms living in the stratosphere, 25 miles above the surface, where conditions approach those of space itself. These organisms seem specifically adapted for surviving in near-vacuum conditions with intense radiation exposure. Some of these high-altitude organisms don't match any known Earth species. Their genetic signatures suggest they might be recent arrivals from space, rather than terrestrial life forms that drifted upward. If microorganisms are regularly arriving from space, they could be continuously introducing new genetic material into Earth's biosphere. Space station experiments have confirmed that Earth organisms can survive extended exposure to space conditions, but they've also revealed something unexpected. Some bacteria actually thrive in space, reproducing faster and developing new capabilities that they don't possess on Earth. It's as if the space environment activates dormant genetic programs designed for interplanetary survival. The cumulative evidence from modern space exploration supports the idea that life is actively traveling throughout our solar system and potentially between star systems. The panspermia process isn't just ancient history, but an ongoing phenomenon that continues to shape biological evolution on multiple worlds. This contemporary perspective on cosmic life transfer helps resolve one of the most puzzling aspects of evolution on Earth: how biological innovation happens so quickly that it defies conventional explanations. Evolution has a speed limit, and life on Earth constantly breaks it. Charles Darwin understood that natural selection requires vast amounts of time to create complex biological systems. Each beneficial mutation must spread through populations, accumulate with other favorable changes, and gradually build the intricate molecular machinery that powers living cells. The mathematics of this process predicts that major evolutionary innovations should take hundreds of millions of years to develop. Yet Earth's fossil record tells a completely different story. Complex biological systems appear suddenly, function perfectly from their first appearance, and show no evidence of the gradual assembly that Darwinian evolution requires. It's like finding modern jet engines in archaeological layers that should contain only wooden wheels. The numbers are staggering. Developing a single new protein through random mutation and natural selection should take roughly 100 million years under optimal conditions. Yet the Cambrian explosion introduced hundreds of new protein families simultaneously in just 25 million years. The mathematical impossibility is so extreme that some scientists have suggested evolution must work through unknown mechanisms that operate far faster than anything we can observe or measure. Consider the complexity we're dealing with. A bacterial flagellum, the rotating motor that propels many bacteria, contains over 40 different protein components that must work together with clockwork precision. Remove any single component and the entire system fails. Evolutionary biologist Michael Behe calculated that assembling such a system through random mutations would require more time than the universe has existed. But panspermia solves the time-scale problem elegantly. Instead of requiring each planet to independently evolve complex biological systems from scratch, cosmic seeding allows worlds to inherit pre-designed genetic modules that were developed over billions of years by civilizations throughout the galaxy. From this perspective, Earth's rapid biological innovations make perfect sense. When cosmic seeding packages arrive on our planet, they don't just deliver simple organic compounds. They bring complete genetic blueprints for sophisticated biological machines that were perfected on countless other worlds. The sudden appearance of complex systems in Earth's fossil record reflects the arrival and activation of these cosmic genetic libraries. Recent research in synthetic biology supports this interpretation. When human scientists try to design biological systems from scratch, they invariably create modular architectures where complex functions are built from interchangeable genetic components. This is exactly the pattern we see in Earth's most sophisticated biological systems. The genetic modules responsible for complex traits like photosynthesis, nervous systems, and immune responses show characteristics of engineered rather than evolved systems. They're too well integrated, too mathematically optimized, and too functionally sophisticated to be products of random mutation and natural selection operating within Earth's limited time frame. If biological systems were designed by cosmic civilizations with billions of years of development time, then the speed of innovation on Earth becomes not just possible but inevitable. Each major evolutionary leap represents the successful integration of genetic modules that were tested and refined across countless worlds before arriving on our planet. This perspective fundamentally changes how we think about humanity's place in the cosmic story. We're not just random products of terrestrial evolution, but inheritors of a biological legacy that spans the galaxy and reaches back to the earliest civilizations in cosmic history. The full implications of this inheritance challenge everything we thought we knew about the nature and destiny of human civilization itself. If the Panspermia hypothesis is correct, then human beings are not just another species on Earth, but the latest iteration of intelligence in a cosmic story that spans billions of years. Every human cell carries genetic information that might have been designed by civilizations that existed before our solar system formed. The DNA in your body could contain biological blueprints tested on thousands of worlds, refined through countless evolutionary experiments, and delivered to Earth as part of the greatest engineering project in galactic history. This perspective transforms how we understand human potential and responsibility. We're not isolated accidents in an indifferent universe, but products of a deliberate cosmic process designed to preserve and spread intelligence throughout the galaxy. The rapid development of human technology, from stone tools to space travel in just 10,000 years, makes sense if we're rediscovering capabilities that our genetic heritage was designed to support. Our drive to explore space, to search for other life, and to spread beyond Earth might not be cultural choices but biological imperatives, encoded in our DNA by ancient civilizations that understood the importance of cosmic expansion. The same genetic programming that led to the development of language, abstract thinking, and technological innovation might also be driving us toward becoming a spacefaring species. Recent advances in genetic engineering suggest that we're approaching the point where we could deliberately modify organisms for space travel, just as ancient civilizations might have modified the genetic packages that eventually became life on Earth. We're developing the ability to encode information in DNA, create organisms that can survive extreme conditions, and design biological systems for specific purposes. If we are indeed the products of cosmic seeding, then we have a responsibility to continue the process. Our destiny might not just be to explore and colonize other worlds, but to serve as cosmic gardeners, preparing new planets for life and spreading intelligence throughout the galaxy. We could be the next link in a chain of cosmic civilization that stretches back to the dawn of the universe. The search for extraterrestrial intelligence takes on new meaning from this perspective. We might not be looking for alien civilizations, but for our cosmic relatives, other worlds, where the same ancient seeding process successfully created intelligence. Every potentially habitable planet could harbor not aliens, but cousins, descendants of the same galactic heritage that produced humanity. The mathematical precision of Earth's conditions for supporting life, the optimization of our genetic code for surviving cosmic radiation, and the rapid development of human intelligence all support the idea that we're part of a designed, rather than accidental process. We're not just random products of evolution, but engineered successors to civilizations that sacrificed themselves to ensure that intelligence would persist throughout the cosmos. This cosmic perspective on human origins suggests that our greatest adventures lie not behind us, but ahead. We're not the end point of evolution, but the beginning of something far more significant. We're the universe's way of understanding itself, carriers of genetic memories from civilizations that existed billions of years ago, and potentially the ancestors of intelligences that will spread throughout the galaxy billions of years in the future. The Panspermia hypothesis doesn't diminish human significance. It reveals that we're part of the greatest story ever told, the universe's ongoing effort to create and preserve intelligence against the forces of entropy and extinction. We are not alone. We never were.